Hello, everyone. Thank you so much for joining us today for our webinar titled The Science Behind a TLC Test. This webinar will be hosted by my colleague, Patrick Fast, who is a technical manager with our Eurofins Botanical Testing in Brea. I'm Amy Knock, and I'll be moderating the webinar today. Before we begin, I just wanted to let you know a little bit more about how the webinar will run. The webinar is being recorded, and the slides and recording will be available to you within 3 business days. A short Q&A session will follow the presentation to answer any viewer-submitted questions that you might have. During the webinar, you can simply submit questions using the webinar sidebar menu. Select the Questions tab, type in your question, then hit the Enter key on the keyboard. And remember, you can submit questions at any point throughout this webinar. That will conclude my brief introduction today. Patrick, I will now pass it over to you. You may begin your presentation.
Thank you, Amy. Good morning, everybody. All of our guests today, we appreciate your taking the time to join us for today's webinar on the science behind a TLC test. I also want to say thanks to our marketing team with Eurofins and to the Brea team for support in this presentation. So my name is Patrick Fast. I'm a technical manager here with Eurofins Botanical Testing in Brea, California. I have about 8 years of botanical identity testing experience in testing labs and manufacturer labs, and I'm really looking forward to kind of sharing some of my experience and what we do on a day-to-day here in our TLC testing. So we can jump right into it, hopefully. Apologies for that. Amy, sorry for the technical difficulty. I'm seeing the slide isn't, maybe if I...
Yeah, there you go. Yeah, now if you want to select share again, it is showing the pointer. So if you want to go back into presentation mode, Patrick.
Okay, I think it's the pointer I was using, hopefully. Sorry to the audience for the technical difficulty here.
Well, otherwise, if you would like me to, we can, oh, now you're back. If you want to try advancing.
Oh, there we go.
Yeah, there you go, thanks.
Okay, first time jitters. Yeah, thanks for your patience to everybody in the audience. I haven't presented a webinar before. So we open the chat for additional questions, and we just hope to answer some of those questions up front. So the first one, what is TLC? Hold on to your seats because we'll get to that one in the next slide if you don't know what that is. So what is a compendial TLC method? That's what we're going to dive deeper into today as a vehicle for this webinar. How do you select a fit for purpose method for botanical identity testing? And what are acceptance criteria? Why are they important in the HPTLC testing method? What are advantages and limitations of testing with an HPTLC method that is compendial? So what is TLC? Thin layer chromatography. That's what TLC stands for. It's also known as HPTLC, which is high performance using instrumentation. The general science behind TLC is that it's planar chromatography, and with botanical identification, you're comparing a sample against reference standards, and using the comparison, observing the banding pattern between your sample and your standards, and using your method, you're going to conclude to the genus, the species, and plant part, or other types of substances for non-plants. So just a general kind of really simplified version of TLC, where the analytical process starts with preparing your standards and extracting your samples according to the method. Here we have one of our fine chemists preparing an array of reference standards at specific concentrations for various methods. The standards and samples that are prepared are then entered into the auto sampler for application onto a TLC plate, which you can see our chemist holding a TLC plate in this image, and that's when the chromatography begins. And so how is TLC performed? What does that chromatography mean in the context of botanical identification? So a compendial TLC method is a standardized method that's validated for a specific source of material. So it's validated for one particular botanical or extract, and using one source of that material to obtain that validation data, which defines the scope of the method. So we have an example of what we would look at when we're talking about the scope of a compendial TLC method. In this case, it's a Melissa leaf, otherwise known as lemon balm. This one's an extract. And you can see there's specifications about what this monograph defines as a Melissa leaf dry extract. Specifically here, you can see that there is hot water or hydroalcoholic solvent that is used in the production of this type of extract. So to meet the scope for this pharmacopoeial method, your sample would need to fall under these parameters in order to ensure that the testing is fit for purpose. So that leads us right into method selection and scope and how to ensure fit for purpose testing. What we need to do that is specific information that will allow us to verify that we select a method that is appropriate based on the type of sample. So the Latin binomial is the genus and species. That's the first piece of information. Along with that, we need the plant part or substance type, if it's not a plant, fungi, algae, that sort of thing. And additionally, the extraction, as we noted before, the solvents and any steps in processing or additional standardization that's required to produce this material. We would want to be aware of all that information so that we know that it fits the scope of the method that we have available. And additionally, a C of A or specification document from either the vendor or your own internal specification that defines what your product standards are, that will help us confirm the testing needs that are required to confirm those specifications. So moving to acceptance criteria and conclusions, these are part of the method. So when we run the samples on the TLC plate and perform the chromatography according to the method, the resulting chromatograms are going to be evaluated, and we use clearly defined acceptance criteria to make the conclusions, whether it meets the acceptance criteria or does not meet the acceptance criteria. The system suitability test is often a component of the compendial method, and that simply verifies the method suitability. The chromatographic system is suitable for this specific analysis. So each time you run the method, you want to confirm that you can accurately analyze your samples, and the system suitability test is what allows us to confirm the accuracy and performance of the chromatography. The parameters that you'll see when we're talking about TLC data is going to be the RF value or retention factor. How far up the plate does the band travel, the color of that band that has migrated up the plate, and the intensity of those bands are all considered in acceptance criteria and how the data is evaluated. The final result based on meeting or not meeting the acceptance criteria is issued as it conforms or does not conform. And on your C of A, in addition to the result, you will receive the information about the method, including the reference to the monograph, the specific acceptance criteria that are used in the monograph, and then the observations that were specific to the sample being tested and the plate images. So with all that information, you could confirm that the observations were appropriate for the chromatography and it was evaluated accurately against the acceptance criteria of the method. So with all these tools we've discussed, we can now take a quick dive into some examples of chromatography using various compendial methods based on different client questions and inquiries that we encounter. So the first one, we have an example where we were looking to confirm the identity of a monk fruit extract. So the method selected for this testing request was the Food Chemicals Codex monk fruit extract identification by thin layer chromatography. Food Chemicals Codex is the pharmacopoeia in this case. And so first we have here just the image of the plate and the lanes 1, 2, 3. We have our standard solution A and B, and then our sample is in lane 3. The lane 1 is our Mogerside 5 reference standard, and lane 2 is the USP monk fruit extract standard. So this big body of text here is the acceptance criteria from the method, and it states that the principal spot obtained in the sample solution corresponds to standard solution A, which is that Mogerside 5 standard. And you can see that the principal spot in lane 3 does correspond to the principal spot in lane 1, so it meets that portion of the acceptance criteria. Second portion of the acceptance criteria states the chromatogram obtained with the sample corresponds to the placement and color of the spots obtained with solution B, which is the USP extract standard. And so we can see 1, 2, 3, and 4 spots that are corresponding in placement and color, and they are increasingly faint as you go up the chromatogram, but they meet the acceptance criteria, and so the result here was conforms. And so that's sort of what we go through every time we do a sample in TLC. We'll walk through this exact process, and that is how our data is generated for our results. So next example from the client, do you test for aloe ferox leaf juice? And in this case, the method that was identified as fit for purpose for this particular request is going to be the European Pharmacopoeia for Cape aloes. The identification section of that monograph contains a TLC test. And so what we have is the first image of 2 images that we'll see, where you can see the actual plate on the left with the dark background, and on the right there in that box is what we'll see for acceptance criteria in this particular monograph method. And essentially that displays the banding pattern that should be observed in your reference solution on the left of that box and the test solution on the right of that box. So when we look at our reference solution here, the upper zone of the chromatogram displays a yellow fluorescent zone for aloe emodin. And in the middle portion of the chromatogram, we'll see a orange fluorescent zone for barbaloin. And we can see in our reference solution on the plate image that it does correspond to those same general areas, colors, and the placement of the bands. So that's serving to show that our reference solution is behaving as expected on the plate. So we can move to analyzing the test solution. And what we see on this lane is a blue fluorescent zone and then the upper middle area, which we see in the sample. And then below that, you'll see a blue fluorescent zone with an orange fluorescent zone directly underneath it. And we see that on the left in the chromatogram. In the lower portion of the chromatogram, there's a bluish white fluorescent zone, which we're seeing at around just under 0.3 RF value on that plate. Additionally, in the acceptance criteria, there is a statement in this particular monograph for additional weaker bands may be present in this test solution. And so if you're wondering why there's only 4 bands on the acceptance criteria, but there's a few extra bands in the sample, that is accounted for in the acceptance criteria of the method. So there's another image and another set of acceptance criteria for this method. And the difference is on the dark background, that's a UV 366 nanometer light visualization. On the light background, that is a white light visualization. And the images are captured in a TLC visualizer instrument. The reference solution on this image has the reference standards are the same. And so we're seeing the alloy modin again, and it is at the same RF because this is the same plate, but the color is violet in this particular acceptance criteria. And the barbaloan is now a brown zone. And we can see that those are corresponding in the sample or reference solution and our acceptance criteria. And moving to the sample itself, just one band in this image for the acceptance criteria, the brown zone due to barbaloan. And you can see that the RF value of that band in the sample is also corresponding to that band in the reference solution, which is how we can confirm by this method that that is in fact the barbaloan compound in that band. And so with all of that, the result is reported as conforms based on having the same bands represented in the acceptance criteria. And we can move on to our next example. Another question, is there ID testing for a lemon balm extract, in this case produced with a 1-to-1 ratio of ethanol and water? If we remember back to that earlier slide, there was that hydroalcoholic extract in the scope of that Melissa dry extract monograph. And so that is what you see we selected as the European Pharmacopoeia Melissa leaf dry extract identification test. And we have our plate image again with our acceptance criteria. And so again, you have the left and the right for reference and test solution. And the first band we're going to look at is going to be rosmarinic acid, a light blue fluorescent zone, and that is present in our reference standard solution. Then there is a hypericide, orange or greenish yellow fluorescent zone, which is present in our standard solution, and a ruticide, orange or greenish yellow fluorescent zone, present in our standard solution. So this standard is prepared with all 3 of these compounds, and they are showing the RF values and colors that are described in the method. So again, we know that our chromatography has been performed accurately enough to evaluate our test sample on this plate. So this one also shows a band for rosmarinic acid in the test solution. Then another blue fluorescent band also in the upper region of the chromatogram. In the middle region of the chromatogram, we see a blue fluorescent zone. And in the lower region of the chromatogram, a light blue fluorescent zone. So our test solution is showing bands that correspond to the acceptance criteria of the method, and the result is reported as conforms. So now we'll move into some more examples. The first three were reported as conforms, and now we're going to get to explore some maybe more complex problems that you could encounter on your TLC testing. So a client has a reishi mushroom powder. They submitted it for identity testing. The compendial test method available is the USP, identification A, thin layer chromatography. And this method is for reishi mushroom powder, indicating it's a crude fruiting body powder, not an extract. So here we have our images on the left. These are derivatized images, and you have your visible light and UV 366 again. Derivatized just means the method has been completed. Each step is all the way. So then from left to right, we have our reference standards and then our samples on the far right lanes. The first standard is ganideric acid A. The second standard is ergosterol, which you can see the arrows pointing to. And the third standard, just before the red box, is our USP Ganoderma lucidum fruiting body powdered extract reference solution. That's indicated as standard solutions A, B, and C in the method. And the evaluation criteria for this particular monograph is the sample should, in general, it's a big body of text and I'll summarize for you, should have bands that are corresponding to some of those ganideric acids, which are in the lower zone of the chromatogram. And you can see there are some bands matching with that USP standard in the sample. But then the additional piece of the acceptance criteria is there needs to be a presence of that ergosterol standard band, corresponding band in the sample. And what we see there is there's no band corresponding to ergosterol, and therefore the result does not conform. And so what do we do when we get a does not conform or out of spec result? Well, we can look at the data, we can share the observations. Next steps with the client involve having a conversation. We want to talk about the report that we've provided you so that we can make sure that there is an equal understanding on both sides of the testing that was performed and of the sample material that was submitted, as well as the information that was submitted with the sample. So going back to one of the previous slides about what information is required to identify the fit for purpose testing, we want to confirm that that information was correct. And sometimes it requires asking the supplier of the material for additional information that wasn't known at the time of submitting. So in this case, in the results of the investigation, we were able to gather some details from the supplier up the chain and learn that the material was actually not a crude fruiting body powder, but it was an extract, which would not be in scope for this compendial method because it's not designed for this specific type of extract, and the acceptance criteria and the method's not validated for this type of material. And so in this case, additional work and investigation can be done to do method development and troubleshooting to determine if there's a better way to go about performing an identity test on this type of material. The next example, we had a question about white willow bark, Salix species, and this was again a crude powder of bark, and the goal was to meet the USP standard for identity. So we selected, upon request, the USP method for Salix species bark powder. And this is identification A, HPTLC, which is just a section of the monograph itself. And so again, we have our images and our lane assignments. From left to right, we have our standards. Salicin is the single band in the first lane that you see, lane 2. The next lane, lane 3, is our Salix species bark standard, and that is standard solution B in the method. In lane 4, what we have is a hydrolyzed version of the Salix species bark extract. And in lane 5 and 6, we have 2 lanes of the sample that are actually, in this case, prepared differently. One in lane 5 is sample solution A, and that is just dissolved in solution. Lane 6, sample solution B, according to the method, is prepared with hydrolysis. And so what we're seeing is this salicin band in the acceptance criteria in lane 5 sample solution A should be of lesser intensity than the standard solution A band for salicin that corresponds where these 2 red arrows are. And so what we're seeing is that in sample solution A, where the arrow is pointing inside the red box, that band is not of lesser intensity than standard solution A. And so that would not meet that particular component of the acceptance criteria, which would lead to a does not conform result. And what we are seeing in the standard solution B, which is our USP Salix extract, where the 2 arrows are pointing, those are salicylate bands that we're seeing not present in the sample solution that is prepared just dissolved in solution. So to summarize, this big red arrow is pointing to a hydrolyzed standard, and the sample solution pre and post hydrolysis, both of those lanes inside the red box, those both look very similar to each other. We don't see any effects of the hydrolysis preparation versus the standard dissolving. And so with this observation, we didn't meet the acceptance criteria, but that observation gives us some data to discuss with the client. We can, again, verify that information that's critical to be submitted with the sample. And we can talk about next steps in the investigation. In this case, we looked into the supply chain, and it was revealed that the material processing is not the fitting for the scope of this USP method that we ran. So while this sample does not meet the USP standard for identity, which was what the client's goal was, we now understand that potentially method development would be required in order to find a way to trace this material back to white willow bark. While it may not meet the USP standard, it's possible that it could be demonstrated to be derived from white willow bark. So our final example is for astaxanthin product that is made from Hematococcus pluvialis algae. It's a red algae that produces the astaxanthin, and the method that was selected for this was USP Hematococcus pluvialis powder, IDA. So this one's less busy. We just have 2 lanes here. Lane 4 is our USP astaxanthin esters from Hematococcus pluvialis. That's the standard solution described in the method. Lane 5 is our sample, and you can see there's 1, 2, 3, 4, 5 bands present in the standard. The acceptance criteria, or the system suitability for the method, describes that there should be these 5 bands. 2 of them are in pairs, and the lowest RF band is the most intense of all. And so we can observe that that is true, and our system suitability passes. And the acceptance criteria of the method states that the sample should contain all the same bands that are observed in the reference standard, and additionally, free astaxanthin band may be observed. In this case with the sample, clearly there's 1 faint band and no other visible bands in the chromatogram, so that one does not conform. The next steps we can take for this sample is, again, just check all the boxes, make sure we have the correct information, make sure that the specifications and C of A's are understood and submitted accurately, and inquire if there's any additional processing of the material beyond what was indicated initially, and if there's any reason that that could alter the TLC fingerprint to result in the data that we are observing. In this case, after the investigation of the results and looking into what the client's goal was, the ultimate outcome with this was truly out of specification for Haematococcus pluvialis algae, and the identity could not be confirmed as being from that algae based on this USP method. All right, so I know that was a lot of information. We're just going to talk through some of the advantages that come up when we're using this type of compendial HPTLC testing. The pharmacopoeial standards methods are validated with robust data that helps us to ensure confidence in every analysis that we perform using that method. The data that's generated when you're using a standardized pharmacopoeial method is readily accepted by quality auditors, regulatory auditors, helps with compliance. The monograph methods have been harmonized by expert committees. There's been a process of peer review and comment and publishing through these authoritative reference sources. The clear acceptance criteria that are a component of every monograph method provide transparency in how we evaluate. As we walk through each of these examples, that is exactly how we would walk through our evaluation on a day-to-day basis with every test that we perform. And that information is again provided on the C of A. So there's when you have the C of A in your hand, there may be questions, but there's a lot of information there to help in the conversation of answering the questions about why is my result a pass or why is it a fail. So the scope of the method, which we highlighted, is a way to ensure that there's fit for purpose testing performed on every sample and that the material that we are testing is the intended use of the method that it has been validated for. And lastly, the standardization that comes with a pharmacopoeial method ensures that from day to day, from different analysts, the results will be reproducible and accurate according to the criteria and validation of that method. So talking about limitations of compendial testing, I can almost smell this slide, the cinnamon and the citrus peels, of course the anise, but these are very aromatic herbs, that two of my favorite aromas, three actually. The limitations, what we listed as an advantage, the scope, it also becomes a limitation when we're talking about compendial ID testing, because that scope is only as broad as the validation data that was used to develop this method, and also only as broad as the materials and reference standards and the inputs that went into developing and validating that method. So while the scope is an advantage to ensuring that the data is fit for purpose, it can also be a limiting factor, and it may not account for all the variability across every region, every harvest, every season, every different type of extract. And so that limitation of balancing all of those factors with the regulatory compliance that is needed and the desire for a standardized method kind of is, that's a story that's as old as the botanicals industry. And, we just want to highlight that as a known limitation, but that's what leads us to those conversations about an OOS result and making sure that we support you through the entire process, from submitting a sample to discussing the results and taking any next steps that we can to continue to support you. So with that, some of those ways we can continue to help and support is beyond the compendial identity testing we've discussed today. There are additional monograph tests, for example, microscopy, that can be used in potentially an orthogonal way to really enhance the results of a single identity test. And outside of compendial testing, we offer method development and validation for additional sample types for any time a sample is deemed out of scope. That's something we would like to explore if it can lead us to another solution. Additionally, we talk a lot about adulteration. We can do development of methods for specific adulterants. Some monograph compendial tests do incorporate acceptance criteria for adulterants or similar species. And then complex matrices beyond just a single ingredient botanical or raw material, just every variation and permutation that exists that we can do method development for. That is something we would like to be able to demonstrate. And just again, highlighting our OS process. When you receive an identity testing result, we want to make sure that we talk through any questions and provide our support, our expertise, and our knowledge moving forward so that you can move through your quality system in a way that you are confident. And yeah, so that concludes everything I had for the presentation today. I know that we have some time for questions. I want to say thank you again to everybody for your patience with the presentation and joining us, and I'll hand it back to Amy.
Thank you so much, Patrick, for your presentation. As Patrick noted, we'll move into a short Q&A here. Again, feel free to continue to submit questions during this session. And if we do not address the questions that you have live, we will follow up with you after today's session. So, Patrick, the first question that has come in here is, what are the next steps for a non-conforming result?
Yeah, okay. Let me think about that one, how to best answer. So yeah, non-conforming result, really that's what we talk about the most when we have the conversation about, any result that if it doesn't conform and, maybe you want more details about why doesn't it conform or how did you determine that? And basically the next steps, we can verify that sample submission that was provided with the sample to ensure that it was interpreted accurately, that there's no new information that would alter the way that the sample should be submitted and ultimately tested, making sure that the method is applicable that was used. Sometimes that information does turn out to be different in reality from what was understood at the time of sending the sample. So that's really the big thing, is making sure that we did in fact perform accurate fit for purpose testing, checking all the boxes there. And then if there is additional information or additional discussion of the process of how that material may be manufactured, we can potentially do development to either extend the scope of a method or develop an entirely new method if it makes sense for a material that has been processed and altered outside of the standard accepted by the method. Let's see, I think beyond that, if there's any other, follow-up questions about OS results, I'd be more than happy to discuss, but I think that's pretty much the main points.
Okay, wonderful. And then we have time for one more question here. So the other one that came in is, what if I don't have complete sample information required?
Yeah, that one comes up, pretty regularly, where it's not always realistic to have every possible detail about a material, and we understand that. And we always want to do our best from the first step of engaging with testing a material and providing whatever expertise or whatever we can to support providing an accurate test. And so if the information is not fully complete, we might be able to infer some information even from a common name. If you say that you have, you have elderberry, and you want to test your elderberry, but you don't have anything other than just the common name. Well, we might be able to make an assumption based on the common name that elderberry is Sambucus nigra fruit. It may not always be the correct assumption, so we want to make sure that in those cases we're transparent about what the assumptions we're making. Additionally, if you have the actual genus, species, and plant part, that would clarify that question. In terms of the extract processing, that type of information, we can attempt to do an ID on just the genus, species, and plant part using the most fitting method for that information. And again, being transparent about possibilities that could result in a non-conform, because that material is not actually fit for that method, or because it's been altered and processed in such a way that we would need to do some more digging and potential method development to really find the appropriate test method. And one other note I wanted to that came to mind on that is the information, while it's required for testing, understanding the origins, quality, and the true specification of that raw material, that is essential for ensuring that label claims are accurate and compliant. And so more than just being able to submit it for testing, you really want to have that information to ensure that your products are not adulterated and that they're effective and safe. And so that's where being an expert in the products that you're testing is going to go a long way in determining the appropriate testing and getting a solid protocol in place. So yeah, I think being confident and knowledgeable about the products that you're working with will be the biggest benefit that you can, add to your approach to product development and finished products. So yeah, I hope that answers the question for the audience member.
Okay, wonderful. Yeah, and I just want to, again, there have been a number of logistical questions that have come into the chat. So again, I just want to remind everyone, a recording of the webinar and a copy of the slides that Patrick delivered today will be available within 3 business days. So in case you wanted to review those a little bit further, just a reminder that we will send those out in email. So I think that wraps up our Q&A session for today. Again, thank you, Patrick, for your expertise and your presentation, and thanks everyone for attending. Have a great day.